Good afternoon. Today we are discuss another topic, that is the plant tissue culture. Yeah, so plant tissue culture, one of the subject. So, right? Is it right? Okay. So, what is the plant tissue culture means? The plant tissue culture is the in vitro aseptic culture of cells, either tissues or whole plant under the controlled nutritional and environmental condition, often to produce the clones of plants. Means this is the artificial provide the all the conditions for the growth of the whole plant. The technique primarily relies on plant cells, that is totipotency, which is the capacity of single cell to express the whole genome during the cell division. So the time means the totipotency is the able to only the plant cells have that capacity, means in a single cell they are the able to produce the whole plant. That capacities have the only the plant cells. That ability we call as the totipotency. The ability of cells to change their metabolism, growth, and development is just as significant and essential for the regeneration of the entire plants. The plant tissue culture, the technology is being widely used for large scale plant multiplication. In addition to being used in the research, they are essential for plant propagation, disease eradication, and generation of secondary metabolites. So secondary metabolites nowadays very important in the plant physiology. The secondary metabolites are economically more valuable. Okay. So now on the diseased free plants also, micropropagation and produce the diseased free plants also. So history of the tissue culture. In 19th century, Herbert Land that tried to cultivate the individual palisade cells from the leaves in knob cells. That solution supplemented with the sucrose solution. So these Heberland to produce the palisade cells. The cells sustained for a month, stored starch, but the ultimately did not. divide. So this deposit space is considered the further uh, undertaking that. So that's why the father of the plant tissue culture since his experiment set the stage for developing the tissue culture technology. Similarly, Roger um, Guthrie is a French scientist. He is encouraging the result with the culturing cambial tissue of carrots in 1933. So, what is the conditions? The choice of medium is based on the type of plant species. Explants are used for culture for the optimal response. All the nutrients required for a plant's proper growth and development should be present in the plant tissue culture media. The macronutrients, micronutrients, vitamins, and other organic ingredients, the plant growth regulators as carbon source, and in the case of a solid medium, a few gelling agents make up the majority of its composition. So all are the we are to provide the micronutrients and macronutrients, some growth hormones also, carbon source. These are the we are added the for the nutrient media. So similarly, hormones level and culture variable like temperature, pH, light intensity, and humidity also plays an important role in the success of tissue culture. So that is the temperature. Temperature is the room temperature is suitable, means around 25 degrees to 27 degrees centigrade. So that is the room temperature is the most suitable for the growth. And pH also, light intensity, humidity. So these environmental conditions is most important for the growing the culture, means the tissue culture. So minerals consist of macronutrients such as nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur. These are the macronutrients, which is very essential. Such as micronutrients also. This example iron and uh, manganese, zinc, boron, copper, molybdenum, and cobalt also. So these are the micronutrients and macronutrients are more essential for the tissue culture. So vitamins are necessary for the healthy and growth of the tissue culture. The vitamins like thiamine and pyridoxine, nicotinic acid, and other vitamins such as biotic folic acid, ascorbic acid, and vitamin E. These are the some added to media formulation. Means when we are to prepare the media, so these micro and macronutrients are added to the that growth media. The plants also require to an external carbon source, sucrose, sugars. These are there. The most commonly used carbon source is sucrose. Other sources are used glucose, maltose, and sorbitol. The pH of the culture medium remains vital as it influences the uptake for various components of the medium and regulates a wide range of biochemical reactions. Most media are adjusted around 5.2, 5.8. This one is the very suitable for The tissue culture media. Next one is the tissue culture media. Most popular media for the in vitro vegetative propagation of various plants in Murashigi and Scoop media. That one will call as a MS medium commonly. Murashigi and Scoop medium. For culturing the either solid or liquid media can be employed. That is the crown woody plant medium and uh, consensus media. These are the plant growth regulators are crucial for determining the development of plant cells and tissues in the culture medium. The most commonly used plant growth regulators which are auxins and cytokinins and gibberellins. The high auxins concentration, which is the favor to the development of fruits. The most commonly used auxins, which are the IAA, means indole acid, IBA, indole acid, NAA, naphthalolic acid, and 2,4-D, that is we call it 2,4-D dichlorophenic acid. The cytokines which are promote the cell division and shoot growth. The most commonly cytokines used would BAP means benzylamine, gelatin, and isoprenols and kinetin. The cytokines are the generally dissolved in the dilute HCl. A clump of undifferentiated cells called as the callus that develop with the auxins and cytokines
So the clump undifferentiated the cells called callus, that is the when we the organs and cytokinins levels are balanced. So some tissue culture vessels, what are the vessels they are to use means they are to use. So another critical aspect in the plant tissue culture, the management of the greenhouse and other hormones, ethylene. So in the closed culture vessels used for in vitro plant growth, ethylene builds up and is often is determinal to the culture. The addition of ethylene biosynthetic inhibitors such as the silver nitrate. So the silver thiosulfates have been shown to increase the formation of shoots. The cultures are grown in what growth room or growth chamber. Humidity, light intensity, temperature must be controlled for the proper growth of the culture. Around 16 hours light to photo period is optimal for the tissue culture, and the temperature around 22 to 25 degrees centigrade is used in the most laboratories. Means these 22 to 25 degrees is the common for the mm, culture. So cool white fluorescent lamps also supply a light intensity of 25 to 50. The relative humidity of 50 to 60 is maintained in the growth chamber. Some cultures are also used incubated in the dark. The cultures can be cultivated in various containers, including test tube, flask, petri dish, and bottle. These are the anyone we have to use for the culture vessels.
The preparation of sterility environment is necessary for effective tissue culture. The laminar airflow food is used for the all tissue culture work. A dust filter and high efficiency particular that air filter are used in the laminar airflow. The hood must be kept stop cells, which can be accomplished by whipping with the alcohol that contains 70% of the alcohol. The surface of the plant tissues naturally contain a variety of bacteria, fungi. Before tissue culture, it is crucial to thoroughly clean the explant since contaminations can proliferate in the culture medium. They also compete with the plant tissues for nutrient differing of the nutrient. The bacteria and fungi can rapidly surpass plant tissues and destroy them. So eggplants are commonly surface sterilized using the sodium hypochlorite, ethanol, and fungicides. When used the filled grown tissues. The type of tissues usually decides the time of sterilization. Leaf tissues requires a shorter sterilization time than the seeds with the hard seed coat. So some tissue culture types. What are the tissue culture types here? Uh, so first one is the callus culture. A callus is an organized mass of cells that develops when the cells are wounded. When the explant is cultivated on the media that promote the development of undifferentiated cells, a callus is formed. The majority of the callus cells are formed with the aid of auxins and cytokines. Means here, mm, when we are to use the cytokinins and auxins, growth hormones are must for the culture.
So when we add this one to develop,
Cells are formed. The cluster-like means group of undifferentiated cells, mass of undifferentiated cells, that one we call as a callus. So using these plant hormones, callus can multiply continuously or to be directed to develop the organ or somatic embryos. That is the callus culture. So next one is the cell suspension culture. Small fragments of loose is the fibril callus can be cultured as the cell suspension culture in a liquid medium. In we are to use the liquid medium only. The cell suspension can be maintained such batch culture grown in the flask for long periods. A portion of callus tissues can be transferred into a liquid medium, and when the subjected to continuous shaking single cell culture and suspension cultures can be cultivated from callus. The growth rate of the suspension cultures, the cells are generally higher than that of the solid culture. Another one is the anther culture. What is anther culture means? Elevated microbes to produce the applied plants is known as the anther culture, or another term, the microspore culture also. The embryos can be produced by a callus phase or to be directed recapitulation of the developmental stages. The characteristic of zygotic embryos compared to traditional breeding method, the microspore culture is unable to the creation of homozygous plants very sharp. Homozygous plants means they have the applied plants only that were similar. mm, There is no recessive character. Means applied character only. So these homozygous plants are useful in the plant breeding and genetic studies also. The breeding technique, plant breeding technique, the homozygous are most important and their vital role in the produce the new varieties. And simultaneously genetic studies also in the molecular level or genetic DNA markers and DNA level. These homozygous plants are more important in that genetic level. Another one is the protoplast culture. So protoplast contains all the components of plant cell except the cell wall. Here without cell wall, that's what we call as a. So protoplasts can be used to create the somatic hybrids and generate whole plants from a single cell. The cell walls of the explant can be removed either mechanical or enzymatic. Means the cell wall here, the cell wall made up of the cellulose. Sometimes pectin is also present, and suberin hemicellulose is also present. It is very hard in structure. Hemicellulose suberin is very hard in structure. Cell wall is rigid structure. So that's why these somatic hybrids there are the regenerate cell. So cell wall which is removed by the mechanical or studies means then we have to sometimes cellulase to one type of enzyme which is digest. So protoplasts can be cultured either in the liquid or solid medium. The protoplast which is embedded in the alginate matrix and culture as solid media, which is the success rate of regeneration of organs. Although the protoplasts appear to be a very appealing method for the regenerating the plants and transferring the genes, that are extremely delicate. Another one is the embryo culture. In the embryo culture, it is the technique in which the solid means isolated embryos from immature ovules or seeds, which is cultured in vitro technique. So embryos are isolated from the seeds, or either sometimes ovules also, match ovules also to develop as a embryos. For species, those seeds are dormant or resistant or prematurely sterile. Embryos culture has been used as a helpful tool for the direct regeneration. In plant breeding program, the embryo culture goes in hand with in vitro control of pollination and fertilization to ensure hybrid production. So this is the most important of the embryo culture. ఉమ్. So embryo culture. So another already discussed that one. The species hold seeds are dormant or resistant and prematurely sterile embryos has been used as a helpful tool for the direct regeneration. Plant breeding progress. The embryo culture goes hand in hand with the in vitro control of population or either pollination and fertilization ensure hybrid production. The additional direct somatic embryos and embryogenetic calyces can be produced from immature embryos. So, meristem, another type of the tissue culture is the meristem culture. Using the apical meristem tip is the possible to produce disease-free plants. This technique can be referred to as meristem culture, meristem tip culture, or shoot tip culture, depending on the actual explant user. The plant apical meristems make good explant for the cultivation of virus-free plants. Hence, this method is usually used to eliminate viruses in many species. So, plant explant. Uh, Here I show to one picture. Here. So this is the tips. Some steps is processing the explant. So, so regeneration methods of plant tissue culture. So that is the two methods are there. One is the organogenesis, another one is the somatic embryogenesis. In the organogenesis, the tissue culture, which is refers the formation of either shoot or root, the equilibrium of oxygen and cytokinins, and the tissues, which is capacity to reach the phytohormones during the culture, or the key factor in vitro organogenesis. If when we add the sufficient balance of cytokinins and auxins, which are the more effective to develop the tissue cultures. So in the organogenesis, can be two types are there. That is the direct organogenesis. Another one is the indirect organogenesis. The direct organogenesis involves the formation of organs directly via a callus phase. For the production of transgenic plants, induction of plants to a callus phase has been used Either the callus is transformed, plants are regenerate, or the primary explant is transformed and the callus is formed and then shoots are cultivated from the explant. It is more important for transgenic plant production. So here indirect method there is no callus means here so callus is formed in the indirect method. From that callus only to develop the complete plants means primary explant is transferred and the callus is formed. So after callus only to develop the shoots and roots. So another direct organogenesis. Direct organogenesis, which is involves the direct bursts or shoots formation from the tissues without a callus stage. The plants are usually propagated by direct organogenesis for improve the multiplication rates and production of transgenic plants, but mainly for the clonal propagation. So here in the indirect method, uh, callus is formed, but the direct method there is no callus here. Directly shoots or buds are propagated to directly develop organ. So that process we call as a direct organogenesis. So another one is the somatic embryogenesis. So somatic embryogenesis is an asexual development process that is produced the bipolar embryo with a closed vascular system from the somatic tissues of plants. Um, it has become one of the most powerful technique in plant tissue culture for the mass propagation, means mass clonal propagation. So, in the rapidly, the number of plantlets are produced from the somatic embryogenesis. So, these somatic embryogenesis may occur directly or via the callus phase for clonal propagation. Direct to somatic embryogenesis is preferred since there is a less chance of the somaclonal mutation. The indirect somatic embryogenesis is usually used in the selection of desired somaclonal variation and for the production of transgenic plants. So encapsulated the somatic embryos are known as synthetic seeds. The somatic after the formation of somatic embryogenesis, we are to use some oxalo acetic acid means sodium oxalate. These are some chemicals are used to pour these somatic embryos. So which is the preserve or which is able to produce the number of days as synthetic seeds. This one to develop as seeds sometime when we are to use the encapsulated somatic embryos. So these synthetic seeds have multiple advantages. They are easy to handle and they can potentially be stored for long period. There is a potential for escape of low cost of the production. So this one is the advantage of some synthetic seeds. So coming to the rooting of shoots.
So, or either field sometimes been or for the transfer to the field to reduce the water loss during the acclimatization. The plants are initially transferred to the green or growth chamber. The relative humidity outside the vessels is often significantly lower than the humidity inside the vessels. Once the plants are acclimatization under the greenhouse condition, they are ready for the transfer to the field. Some what are the advantages in the greenhouses? They are conduct the potency and nutrition metabolism. Division is a differentiation and preservation of the plant cells. Another advantage is the morphogenesis and plant regeneration from individual cells or tissues through the organogenesis or somatic embryogenesis. That is the third one. The variation were generated through in vitro culture. So fourth one is the evaluation of applied through the anther and pollen culture and sometimes ovule culture also. Where the hybridization program through the ovule ovary embryo culture to overcome both pre-zygotic and post-zygotic sterility mechanisms. So another one is the micropropagation of plant materials. In the micropropagation here, the tissue culture only we have to conduct these micropropagation. In vitro selections of mutants tolerant to biotic and abiotic stresses. In vitro culture and secondary metabolites, biosynthesis, plant genetic engineering using the DNA transfer and in vitro culture techniques. These are the some advantages of the in vitro technique. Some disadvantages that is the labor intensive and expensive process. Another one is the vulnerable to many environmental factors. These are the some tissue culture. Okay. So another topic is there, that is the anther and pollen culture, ovule ovule culture also. So already we have to discuss that one, anther culture and pollen. Yeah. So what is the anther culture here? That one.
In the anther culture, here in the that microspore culture also will called as a microspore culture also. Mm, okay, so wait. The anther culture means that is the anthers are cultured on growth media. Sometimes the microspore only we have to separated microspores from the anther, then culture on the nutrient media or sometimes growth media. Yeah, the technology to clone our plants are producing genetically identical plants. The existing part is that any part of the plant can be used to process to regenerate the whole. You know that is the called as a totipotency. Listen, so in the gametic number of chromosomes, which is a number, may not be equivalent to X. Another one is the application means wait. So what is the pollen culture? The pollen is a microspore produced by the seeded plant and appear in the form of the dust. They are extensively used as an explant to produce the applied plants. So during this anther culture, the flower buds are collected, surface sterilization, and anther lobes, which are dissected. Then the anther lobes are squeezed using the scalpels in a small tube or container, the containing nutrient media to collect the microspore. The microspores from callus to the nutrient media, which develops into applied plantlets when grown through the embryogenesis or organogenesis technique. The ability of pollen grain to induce plant development was the first realized by Philip. So mm, that the ginkgo biloba is first introduced with the ginkgo biloba. So is the maybe different basis on the plant species working on that anther collection from the flower buds is a small beaker containing 100 ml of the liquid media, that is the MS or niche media. Is any media we are to use. The process anther again the side of the beaker is to squeeze out pollens. The homozygized anther are in the filter to nylons used to remove the anther tissues, which is the debris, maybe any debris or anther wall. Yeah, the centrifuge, the pollen suspension at 500 to 600 RPM per minute for 5 minutes. The discard, the supernatant, and they kept the pellets containing pollens. Then the suspend, it is the liquid media and wash, it is twice by centrifugation, repeat the process twice. Keep it out of 2.5 ml of the pollen suspension and spread in the petri dish, the soft agar medium. You can also use in the liquid media to grow pollens. Incubate culture here 27 degrees Celsius under the low intensive of light, full light. After 30 days, you will observe the embryos that can regenerate the plants. So these are the I show to some picture here. These are the some pollen grains are there. So this is the some pollen grains here. You can see. So So another the production of applied plants is one of the major option. It because conventional or traditional breeding method are the time taking. They are take many generation of breeding, inbreeding or either product back class. So another use that is the transformation. When the use of producing the transgenic plants, it can be less time consuming. Another one is the crop improvement also. The pollen culture. Okay. So pollen culture. Uh, Use some embryo employed to enhance the quality of vegetable cereals, crops, pulses, another sweet pepper, watermelon, the used. Some advantages. What are the advantages here? This is the simple technique and require the minimum time and able to period.
So, variety of answer subject to cultivation of to exhibit, which is the responses by relying on the pollen culture, the possibility of and callus formation of the anther wall eliminations. So this is the technique for the pollen and okay. So this one is the
అన్నారు అయిపోయింది ఇక పోదాంపా ఇంకా 5 నిమిషాలు అయితే అయిపోతుంది Saturday, Sunday. Ah, okay. Okay. Good evening. ఇప్పుడు ఉంది పోర్షన్ ఆఫ్లైన్ హాఫ్ ఆన్లైన్ అది యూజ్ చేసి చెప్పిన ఇది చెప్తాను ఒక ఎఫ్ఐఆర్ చెప్పిన అది ఆఫ్లైన్ చెప్పిన అదే బాగా తర్వాతగా కలిసి ఉంటుంది ఏ ఇలా అందా మీరు వస్తుందా ఇలా కొట్టేసి అయిపోతుంది దాగురు లెవెల్ కాలే కాలే ఇంకా